So today we're joined by Nick Pugh, and he's going to tell us a bit about his research at Anglia Ruskin.
Okay, thanks very much, Kirk. So I've got a number of research projects that I'm developing at the moment. But I think what's my major line of research at the moment is in the role of zinc in platelet behaviour. So I'm very interested in zinc in a number of different aspects. So zinc, many people won't probably acknowledge this or realise it, but zinc is a very important biological cofactor in many biological systems. So it's an intracellular messenger in a number of cell models and probably many more that we don't know about. So in mast cells, monocytes, other types of cells, you get changes in zinc concentration in response to agonist stimulation. We've shown that as well in platelets as well. So when we stimulate platelets with a number of different agonists, principally CRP, then you get increases in intracellular zinc concentration. And we've done some work, published it last year, on how we think this this change in zinc concentration is correlated with platelet behavior. So you're getting a signaling leading to intracellular zinc responses that lead to cellular behavior, which is consistent with zinc being an intracellular messenger. So that's probably the most important part of my work at the moment. That's what I'm funded to do at the moment. But I do have a number of other research sidelines that are going on.
That's really interesting, and clearly there's a lot of work that's left to be done to fully understand zinc. I just wonder if maybe you could take us back a little bit to kind of like how you first got interested in zinc. What was the first moment that you thought, "Ooh, let's have a look at this"?
Yeah, I can tell you exactly. I think so. I used to work in Richard Farndale's lab in the Department of Biochemistry at Cambridge University, and that's not it's not particularly a platelet lab; it's a collagen lab. So they develop and they make the CRPXL that is then sold to all of the platelet research labs to do GP6 signaling. So one of the things he was doing at the time, and I wasn't particularly involved with this, is they were generating recombinant proteins as I think binding substrates for different cells, or generating recombinant proteins to see how they bound to collagen. It's not platelet related, but there were aspects of this that were sort of platelet related. So one of the things they were doing, they were finding it hard to get a certain type of protein. I think it was a von Willebrand factor recombinant protein. They were finding it difficult to get this generated, and one of the questions they had was whether it was actually folding correctly. Now they had these histidine tags on the recombinant proteins, and one of the ideas they had were if they add zinc to these proteins as they were generating them, as they were generating the recombinant proteins, that may well help with the stability of the recombinant protein. So at that point I said, well, you can't do that, because if you do platelet work, then you'll be putting zinc on the platelets, and that's obviously going to have an effect. Nobody seemed to really clock this, and I thought about it as well. I thought I had no idea why I knew that. I think this went back to a reference I'd read a number of years before, and I did some manic reading to try and find out where I'd got this from. I still haven't found out the exact reference I found, but looking back at that, there is zinc research going back to the 80s or 90s, where groups have taken zinc, put them on platelets, and seen platelet activation.
That's great. We were wondering, are there any techniques or that you use in your lab that you think the society members should incorporate more into their research?
Well, one of the things that's done a lot is in vitro flow assays. And now I know a lot of people do this in the field, but it did annoy me, and still does a little bit, that people didn't really quantify their results very well at all. At the end of a flow run, 5 minutes, they would sort of see what the surface coverage of platelets are like on there. And the big problem I had with that is that you can have a really good surface coverage, but that's not telling you anything about what the platelet's really doing. I mean, a good surface coverage means the platelets are not activated at all. It means that they're basically rolling, or they're stuck on monolayer of platelets on the surface. Whereas if you had a really good activated platelet phenotype, then you can have a reduced surface coverage. You can have big thrombotic lumps. So I put a lot of work into quantifying platelet adhesion under flow conditions. And we came out, when I was in Richard's lab, with a number of parameters of platelet behavior that are really, really easy to obtain from simple flow experiments. So endpoint parameters where you measure the thrombus, you're basically measuring the height, the ZV50 value we called it. We find that's very, very good at comparing results from different laboratories. It gives you a metric that you can compare with other people. But the thing I was most excited about was my measurement of platelet mobility. So many people will track platelets rolling across von Willebrand factor. I had a population-based approach where I'd look at all of the platelets and see whether they were in motion or adherent. And what we showed in the publication that came out a few years ago, that this was very, very similar to a platelet aggregometry assay, where you can see platelets activated and sticking together. But we were doing it in a system that's more physiologically relevant, with von Willebrand factor adhering to collagen. So, I mean, this is the one area that I see papers coming in, I review papers where they say, the surface coverage is this, or the fluorescence is that sort of thing. And I think, with a very, very little work, probably with the images you already have on your confocal microscope, you can get some real important information about the changes in platelet behavior as a response to whatever treatment you're doing.
So you mentioned that your research is funded, and as we know, there are several organisations in the UK that fund research, and we're wondering who funds the work that you're doing.
But I'm very, very fortunate, very lucky, and very grateful to be funded by the British Heart Foundation, who funded my first grant, which employed a great postdoc grant. So BHF is where I've been to for all of my successful research funding. What I would say as well, for any sort of new researchers coming into the field or coming into research, one good idea would be to go for small pots of money to start off with. So I've known people who have come into their first job as a lecturer and applied for a £500,000 BBSRC grant. There is no way that anybody is going to get that funded at that stage of their career. So look for small pots of money. It's something that Kirk and I did when he was here. We applied for the Wellcome Trust Summer Studentship Award, which is about £2,000 worth of money. It's very, very little, but it pays for an undergraduate student to come in and do some research in the lab. So yeah, small pots of money are a great way to go. They're not that difficult to get hold of, even if it's just a conference application. Get a couple £100, go to a conference, you got it on your CV, and money makes money. The more evidence you've got on your CV for generating a bit of income, the more likely it is you're going to get some more grant income after that.
So Nick, I think the first time I met you was at a conference, and then the second time I met you was when I came to interview for a job with you, which was actually my first postdoc out of my PhD. And I know that was your first research grant. So how did you manage that kind of transition between being a postdoc yourself to kind of being a PI, and kind of the extra steps that you've kind of taken since then?
Yeah, that's an interesting question. Yeah. So, I mean, I was a postdoc in Richard Farndale's lab in Cambridge for the best part of, 8 years, I think I was there. So I was there for 1 and a bit program grants. So when it was coming to the end of the second one, it was not even far off. It was about 2 years before the end of the grant. And I thought, I best start thinking about what to do. I was getting on a bit. I can't be a postdoc forever, sadly. So I was looking at other jobs to go for. And obviously I wanted to stay in academia. I wanted to be a research scientist. But then to do that, you need to be associated with a university. So I'd applied for a lot of different universities at the time. I think I applied for Cambridge at the time. There was no way they were ever going to employ me as a lecturer. Obviously, I was starting my career, no lecturing experience, or very little lecturing experience. But I was quite lucky that an advert came for a biomedical science lecturer at Anglia Ruskin University, which is just across the road from Cambridge University. So it's an old post-1992 university, so an old polytechnic, now a proper university that is in a very early stage of its development, going from basically a teaching-centered university to one that is more research-based. So I applied for the job there just for the experience, really. That's really what I was thinking about. I'll go there, I'll get a bit of experience, then next year when I have to do this, I'll be a bit better at it. And then to my great surprise, they offered me the job, and I had to make a decision now. Do I stay in research, doing good research in Richard's lab for 2 years, or do I make the jump now? And I decided, it's just a stupid no-brainer. I have to make the jump to what is essentially a tenured position where I'm going to be employed, for the rest of my career if I wanted to be. So that's a decision that had to be made. I can't stay as a postdoc. I have to go for the first major, tenured role you get. It's a university back in 2013 that was not a strong research university. It was, as most post-92s are, Amanda will probably know, it's mostly a teaching university, which does an occasional little bit of research when they can do. And that was what the situation was when I first got there. I set up my first lab, and by that I mean I borrowed a spectrophotometer, I borrowed a plate reader, and I set them up in the corner of the microbiology lab. So we had a microbiology lab. It's the only place that was containment level 2, so it's the only place that I could get permission to do any work with blood. I had to set up a first aid room where I could actually take blood, and I had to get all the ethics done for this. And the ethics were, it was a traumatic experience going to a university where no blood is taken, and you say, well, I want to take volunteers' blood. And, the alarm bells that goes off in the minds of the people running the university was absolutely incredible. It took months for me just to get permission to do what we were doing every single day, day in, day out in Cambridge University. So that was a major step for me. And like I say, we had our basic research lab set up. We had huge numbers of undergraduate students who were coming along doing projects with us. So we set up, just then doing some basic platelet aggregometry in the lab, just chucking on some chemicals, seeing what would happen. They would get their undergraduate project, and that was fine. And it was around about this time that I was trying to write up for grants. I actually was very proactive with this. So before I actually made it to Anglia Ruskin, I was writing up the zinc grant with a view to getting it submitted when I was there. So I was really on the ball, really doing it as fast as I could. And I got, absolutely mortified and dejected when it got rejected for the first time, which looking back is just ridiculous. Everybody gets their grants rejected all of the time. And I was of the opinion that it always gets rejected the first time. You have to improve it. A second time, it will normally be accepted, the second time round. If it is going to be accepted, it will be done the second time round. So I got grants submitted, rejected, and then eventually accepted. And that's when we underwent quite a stringent recruitment process. Nobody good applied, so we had to take Kirk, and we had a couple of good years doing zinc work. You're talking about the transition between postdocing and a lecturer. So what I'd done when I was a postdoc in Cambridge, I got as much experience as I could with lecturing and with teaching. And one of the best things I did was, it just seemed so trivial at the time, I did a sort of 5 half-day course called the Teaching Associate Program at Cambridge University. This is just to get you a very small certificate qualification saying, yeah, Nick's done a bit of teaching. But that proved to be so useful to me because, when Anglia Ruskin were looking through, 100 CVs of people, they said, well, who's actually got teaching experience? Oh, this guy, he's done a course in it. That puts my CV higher up the list. So I'd recommend anybody wanting to go on an academic career, actually put that bit of effort in. Don't think it's a waste of time. It's something that said, I'm committed. I've done something, I've got a qualification, and it got me registered with the Higher Education Academy as well. So I became an associate member of the HEA. And all of these things count when it comes to, getting a lectureship. After that, it comes in, what's their research like? How much money can they get in? Have they got a history of generating income? And if they've got little bits of money here or there, say, well, they've had a go, they know what they're doing. They've generated, £2,000-3,000 on conferences, on small applications. That puts their CV higher up the list. So make sure, for any ECRs, make sure you actually engage with this part of the process. Trying to get a bit of money in, just to demonstrate that you can get money in, that you are fundable. And that really helps, I think. So since I started at ARU, one of my major aims is to try and make the place a better research institute. So I applied for deputy head of department positions as the research lead, and I luckily got one of those positions. And I had to reapply for it as we amalgamated our schools and changed our structure. So I'm now deputy head of the School of Life Sciences, and that my remit covers business generation, income and research, and postgraduate students, and ethics, and loads of other stuff. But what I'm trying to do is make us a better research university, and a lot of that involves carving out time for the academics to do some research. So a lot of what I've been doing recently is setting up labs, trying to get things working, setting up things like the Human Tissue Act, so we can actually store human tissue for more than a few hours. Getting things like tissue culture sorted out, stuff like that. So, a major part of my role recently, when I should have been concentrating more on my research career, has been trying to make the university as a better research institute funded. And what I would recommend, or recommend everybody, me as well, thinking about commercial engagement as much as you possibly can. And that's a big thing we're pushing out here. There's less money for all of the research groups from government, from charities, from European funds as well at the moment. So think about how external companies could help with your research plan. So we've got a lot of people who are getting funds from small biotech startups to do a little bit of research. On the back of that, they're following some of their own research interests. But what some of our guys are doing are realizing that it's becoming less of a world where you've got your idea, that's what you work on for the rest of your career, and it's moving more on to what do other people want me to research. That will be my focus on my research.
So thanks, Nick, for sharing your transition and how you set out on your own. So I suppose from my own experience is it can be quite daunting setting up your own lab without any research money. So I found a lot when I was applying that and I don't know if you found that or the situation now is, there's very limited startup funds on offer. And I was just wondering if you've got any advice for anyone looking to make that transition on how they get by in the earlier years.
Yes, great question. Great question that I know quite a lot about, having been through it actually. So when I started at Anglia Ruskin, I asked the question, like you, I imagine, did, like everybody does, what is the starting package? Now, how much, how many thousands of pounds would I expect to get from you just to start my research here, and they said there is no starting package. The actual starting up of the lab is really, really difficult. And one thing I did actually, which was really helpful, and what all ECRs should be doing, is generating your collaborations. So you'll obviously have a good collaboration, hopefully, with the person who is a PI of your previous lab. Stay with them, develop the relationship with them. If you can go into their lab, start using some of their equipment. Yeah. So when I started, when I got my first grant, I had on there a number of collaborators. I had Stuart Sage, Martin Mayer-Smith, Matthew Harper, Richard Farndale. I had loads of whoever would actually write me a letter saying, yes, I support this research. Okay, and that's a good thing for a number of ways. It gets people, it gets you noticed by the people reviewing the grant that this guy has got a lot of people behind him, so it's less likely to go wrong. These people all agree with the project, it's likely to work okay. Okay, so collaborations are absolutely key. Get your letters in, all your ECRs, make sure people support you and support your research as much as you can. It's going to put you in a much better position. Okay,
so lots of great advice from you, Nick. I guess maybe if you could distill it down into one, your top tip for early career researchers, what would that be?
What I would say is get yourself noticed in some way, obviously in a positive way, presumably, but Richard Farndale once told me there's no such thing as bad publicity. As soon as you get yourself noticed, people will remember your name when it comes to when they're coming to review your grants. If you're just a name nobody's ever heard of, you get a grant on your table to review, it's less likely you're going to sort of say, well, I'll fund this. If you know who it is, they're doing good work, the sort of thing they're doing, then it's going to put you in a much better position. Get yourselves noticed, for example, like what you two guys are doing. Obviously, you're working closely with the Platelet Society, doing these great videos, working with the ECR stuff, fantastic thing to do. You guys are now noticed. It'll be much, much easier for you to get grant funding, I think.
Thanks so much, Nick. That was really, really interesting. We really appreciate you joining us today and sharing your research and your research journey with us.
Thank you very much for having me. It's been a great experience. I really enjoyed it. I think this is a great initiative you're doing. I think you should do more of it. I think it's fantastic. Thanks very much.
Thank you. And join us next time on Clot Chat.